Unit 1, Life Processes, Scientific Method, Tools of a Biologist. We'll skip the graphing and data tables. We will have a separate video about graphing. Biology, the study of living things. Organisms, all living things are called organisms. They have certain characteristics in order to be considered a living organism. A, made of one or more cells. B, displays organization. So you learned about this in 7th grade science. A bunch of cells working together make up a tissue, which makes up organs, which make up system. For example, in our body, that's how our organization is set up. Grows and develops. All living things have to grow and develop. They have the ability to reproduce. You will see this as a Regents question often. What's the one life characteristic that a living organism does not have to do in their lifetime? So that would be reproduce. Responds to stimuli. You walk outside, that bright sun makes you close your eyes. You respond to that stimulus, the sun. Requires energy. Energy is in the form of a molecule called ATP throughout our body. Our bodies require this. We all maintain homeostasis, which you'll learn about in a second. We adapt and evolve over time. And there's some pictures of some living organisms. This is a red blood cell, a white blood cell, platelets. This is showing like a bacteria, like a single-celled organism reproducing. Life processes. Homeostasis, this is what I was just mentioning. It's the condition of a constant internal environment. We want everything to be happy. You want a certain temperature. If it's too hot, you sweat. If it's cold, you have little goosebumps trying to keep the heat inside your body. Nutrition is another life process. Every organism takes materials from its environment and changes them into forms it can use. You have ingestion as part of this, taking the material in, digestion, breaking down the material, or egestion, or elimination when it exits. We also have transportation. Transport. It's the process by which substance enter and leave cells and become distributed within cells. In our body, the transport is through the bloodstream. In a simple organism like a one-celled organism, it just, or food or whatever it is, the substance may just diffuse into the cell. It's just one cell. It can move around and stream in the watery filled inner part of the cell, the cytoplasm. But that is transport. The next life process, respiration. I mentioned that everyone requires energy. Respiration is how we get the energy. It's the process of releasing that chemical energy. It comes in your body through carbohydrates and a simple sugar, and then it's released as ATP. There's a chemical reaction you will learn this year. Now, our body's kind of cool. There are two ways to do this, and we do both. Not all organisms do this, but we do aerobic respiration. That requires making ATP with oxygen. And we also do anaerobic respiration. That's without oxygen. Synthesis. Organisms combining simple substances chemically to form more complex substances. I need to move this up. I'm going to do that when I get on the other slide, but synthesis, think of to make. Assimilation, incorporation of the material into the body. So once you've made it, say you cut your finger, you have a scab, your body makes a protein to make the scab. That's called synthesis. Then you assimilate it. It becomes part of the body. Growth, the process by which organisms increase in size. Excretion, the removal of harmful metabolic waste from the organism's body, urine, nitrogen. So what's toxic for us is nitrogen. It comes in on protein. We do not store this. We want to excrete it as urine. Regulation, all the activities that help to maintain an organism's homeostasis. So our bodies are constantly regulating, overall regulating. Reproduction, the process by which living things produce new organisms of their own kind. Asexual is one parent, sexual is two. When you're a product of sexual reproduction like us, you look like a combination of your parents. When it's asexual, the genetic material is identical, so it's like a clone. They look just like their parent. That's asexual there. There's an example of sexual. Metabolism, all the chemical reactions occurring within the cell. All the reactions going on, refer to that as metabolism. You may have been told you have a high metabolism. It just means those chemical reactions are occurring quickly. The tools of a biologist. Now we will show you these when we have our classes, the synchronous classes, but you have a compound microscope. That's what used in 7th grade. There's a phase contrast microscope. It allows details within living specimens to be seen without staining. A lot of our videos will have this. We do not have a phase contrast microscope at the high school level. A stereo microscope, we do have some of these here. They're used to study external parts of a specimen. You would use those when we do dissections. Electron microscope uses electron beams. It can magnify more than 250,000 times. This microscope is super cool. I will show you pictures from one. My college had one. You wouldn't really see that at a high school level. A cell culture. This is placed in a cell. The cell is placed in a dish with nutrients where it can reproduce. The dish is called a petri dish. I will show you some of these. An ultracentrifuge, we have one of those. It's a device that can separate parts of the cells based on different densities. If you've ever had blood drawn, they pull your blood. You sometimes see the nurse or whoever does it put it right in a test tube. Sometimes you see them put it in a machine and it spins. It separates the parts of the blood based on density. It's referred to as a centrifuge or an ultracentrifuge. Chromatography, the separation of a mixture by passing it in a medium based on solubility. I will show you some apparatus for chromatography as well. And then the last tool, electrophoresis or e-gel, the separation of DNA based on density. This is the DNA testing. We have this equipment here as well I will show you during our live synchronous teaching. The scientific method, we all use this. It's a universal approach to solving a problem. You do this all the time. On the left, I wrote each step. Here's a real life example. State the problem. So you wake up, you have a sore throat. Gather some information. You look in the mirror. Oh, it's red. It's irritated. You have a temperature. Maybe you end up not going to school. You end up at a doctor. Form a testable hypothesis. If my throat is red, fever, then it is strep. Maybe the doctor makes that or the nurse. Design and perform an experiment. That would be a throat culture. Draw conclusions based on the data. Now, positive, it may come back. If you could take a pencil in your notes, I could not figure out how to do this, but could you bring a pen or pencil, and if it's positive, you accept the hypothesis. So draw a line here. I color-coded it. And then you would go on to the next step, publish or act on the results. If it's strep throat, they're going to prescribe an antibiotic. In this case, it's negative. In this case, you reject your hypothesis. Please draw a line between negative to reject, and then you actually are going to go back up here and collect more data. You're going to form a new hypothesis. You're going to run through this again. There are some people, like, they're doing research and their hypothesis just isn't what is proven. So they repeat the experiment over and over. We'll skip this page for now. Your teacher will use this when you're going through the notes to do another example of a scientific method right in your notes. So when we're talking about science, you always have a problem, a scientific question. What do you want to investigate? It's often written as, what is the effect of temperature on fish growth? There's an example. A hypothesis is that educated guess, the expected outcome of an experiment based on their research. Always written as a statement, underline that. It's not a question. There will be kids that get all the way to the Regents and they're still forming a hypothesis like a question. Data, information gathered from observation. You have quantitative and qualitative. Quantitative is measured, like your metric system when we do labs and we're measuring. Qualitative is not easily measured. It's more descriptive. The experimental design. you can only test one variable at a time. You can only test one variable at a time. The constants are the factors that stay the same. The variable is the factor that changes. So let me give you an example. If I fertilize my tomatoes, then they will increase in growth. Okay, so I'm testing one variable, fertilizer present or not fertilizer. Okay, I have two pots, same size pot, same soil, same water, same sunlight, same plant, same size of plant. Pot A gets no fertilizer. Pot B gets fertilizer. What are some constants? The soil, the pot, the type of plant, the size of the plant, water, sun. The variable. What changed from setup A to setup B? The fertilizer. The control group, I'm going to skip down here because I'm giving you this example, an experimental group that doesn't have the variable. So was that pot A or pot B? Well, it was pot A. It did not have fertilizer. Why do we even do that one? Why do we even set it up? Because it's used for a comparison. It is the control group. So in drug trials, they have placebo effects. So in this example, I use plants, but let's say you were testing something to cure cancer. Then the control group gets the placebo, a sugar pill. So it's like a little, because the control group doesn't, the people in a controlled study do not know if they're in the control group. So they're taking the pill that looks just like the cancer pill, but it just has sugar. It's fake. It's a placebo. The independent and dependent variable, the value that the scientist manipulates to see what's affected. It's what circle the I and write I control. I control it. I'm the experimenter. What did I control in this example I gave you? Presence of fertilizer. So the independent variable would be no fertilizer or fertilizer. The dependent is the measured outcome. What was the measured outcome? The plant growth. And the last thing for this first unit, note scientific theory versus a law. A hypothesis that is well supported by evidence is a theory. A law is a statement that describes like a relationship in nature that's always true. Think of gravity. And that's it for Unit 1.